Thanks everybody for coming to this special lecture today by Byron Caughey. Got to get the vowels right. Byron was born in Peterborough, New Hampshire. Went to high school at Westtown School in Pennsylvania. Went to Colorado State University and got an undergraduate degree in chemistry. And then came here to UW-Madison to get his PhD in biochemistry. He postdoced at Duke University, and then in 1986 went to the NIH Rocky Mountain Labs in Hamilton, Montana, where he has been ever since. He's here in Madison the last couple of days because he and his wife in particular, has donated a new piece of art for us called Ubiquitous Migration of Pathogens. You can see it out in the South Atrium. That's also why we're having a reception, so that everybody at the Biotech Center in the Genetics Building can come and meet Byron and meet Pamela at 3 o'clock, so I hope you'll stay for the reception afterwards. Today, Byron's going to talk with us about exploiting the prion-like properties of misfolded proteins to diagnose neurodegenerative diseases and to quantify etiological biomarkers. That about right? Please join me in welcoming Byron back to UW-Madison.
So thanks, Tom, for the opportunity to tell you about our work last night in the lay talk. I focused on how corrupted protein aggregates can act as pathogens, just spreading within the body and between individuals in some cases. And today I'm going to focus more practically on our abilities to, or attempts to, exploit this prion-like self-replicating properties of various misfolded proteins in order to better diagnose neurodegenerative protein misfolding diseases and to quantify the levels of these disease-causing protein aggregates as biomarkers. And so, let's see. Just to remind you, as you know, in order to function properly, proteins have to fold up into their active three-dimensional states. And sometimes along the folding pathway, or even after the proteins are folded, problems can occur, allowing them to aggregate into disordered or even ordered structures that can coalesce and reshuffle into highly ordered, sort of generic misfolded protein structure called an amyloid fibril or plaque, as these sort of ribbon-like structures bundled together into big bundles. And normally we have quite effective protein quality control mechanisms that prevent this from getting being a problem, prevent these species from accumulating, but sometimes things spiral out of control. And you can get massive accumulation shown here in the brownish red here in this cross-section of a scrapie grain that's infected with a prion called scrapie. And so in this case, the protein that's accumulating is called PRP, or prion protein. But there are many other proteins, as you probably know, that can do similar things, accumulate, and give rise to a number of very important diseases. And then there is, of course, as I mentioned, prion protein, or PRP, that gives rise in humans anyway to Creutzfeldt-Jakob disease most commonly, in animals that cause chronic wasting disease in deer and elk, present here in Wisconsin, mad cow disease, or BSE in cattle, and so forth and so on. Beta amyloid is a protein that whose misfolding is central to the pathogenesis of Alzheimer's disease, as is the misfolding and aggregation of tau. But tau alone aggregation can give rise to a number of other types of dementias and neurodegenerative diseases as listed here. Alpha-synuclein is a protein that can misfold and give rise to Parkinson's disease, dementia with Lewy bodies, multiple systems atrophy, and so forth and so on. And there are other proteins as well involved in very important protein misfolding diseases. So collectively, these diseases, protein misfolding diseases, represent huge burdens of disease and great expense to our economy, especially as we move ahead. Currently, for instance, Alzheimer's disease alone affects 5 million Americans. That's 11% or so of people above the age of 65, at an annual cost already of $259 billion per year, and will only be increasing as the population ages. And so most, if not all, of these protein misfolding diseases involve the deposition of fibrillar amyloids that can propagate, at least within the host, like prions propagate as misfolded proteins. And several of these, at least, are experimentally transmissible from patients who have these diseases to experimental animals, including, of course, prion diseases, which can be transmitted naturally as well as experimentally, but also Alzheimer's, various tauopathies, synucleinopathies like Parkinson's disease, and so forth. And so the big question that's before us, and we really is not answered yet, is even if these are transmissible experimentally, are they ever really transmissible to a degree that should concern us in real life situations that might exist in clinical situations especially? I mean, not really in terms of casual contact, but perhaps iatrogenically through medical procedures. So I discussed these sort of issues a bit last night. I'm not going to go through them today. What I really want to focus on today is this other big problem relating to these neurodegenerative misfolding diseases, and that is the difficulty of clearly diagnosing these diseases, especially early on in the clinical phase of disease, or preclinically for that matter. I mean, it's so difficult because of overlapping clinical science and so forth, especially early on, that it's very often misdiagnosed. In fact, Parkinson's disease, for instance, is often misdiagnosed half the time, especially by GPs, and maybe 20% of the time even by movement disorder specialists. So it's really a big problem when you're trying to figure out how to handle these diseases. And the final diagnosis, unfortunately, often depends on post-mortem examination of the brain, which is too little too late. So our goal, and the goal of a number of other labs now, is to improve the early diagnosis of these diseases by exploiting the amplification power that's inherent in their prion-like seeded propagation mechanisms that they use to propagate within and sometimes between individuals, in order to develop ultrasensitive tests that can be applied not to brain tissue necessarily, but to more diagnostically accessible tissues and fluids like cerebral spinal fluid, maybe someday blood, skin, that sort of thing. So how are we going about this? What's this really look like? Well, this cartoon is of this self-replicating seeded polymerization mechanisms that prions and prion-like misfolded protein seeds of various sorts have, and that is this ability to grab onto the normal form of the protein in its monomeric state, induce a conformational conversion to the prion-like conformation, do this over and over and over again, grow the fibril, then there's some fragmentation process that happens to generate more seeds than you had, and you can go around and around this cycle indefinitely until to the point where in tissues of the body, these aggregates accumulate to pathological levels. So in order to exploit this sort of replication cycle diagnostically, we have devised this sort of experimental testing scheme here, developed initially by Ryuichiro Atarashi, former postdoc in my lab, and Jason Willem and Christina Oru, postdocs in my lab as well. Ryu is now a professor at Miyazaki University in Japan. But anyway, what he initially figured out could be done is to take a 96-well plate and mix together a tiny amount of your test specimen, diagnostic specimen of some sort or other that may or may not have prions, incubate it with a vast excess of recombinant monomeric substrate protein of your choice, and an amyloid-sensitive dye, thioflavin T, that lights up when it binds to amyloid. And so if your sample has these sort of seeds in it, you can see that very rapidly in this sort of reaction, you put this plate, by the way, into an instrument that shakes it and incubates it and reads fluorescence periodically, usually every 45 minutes or so. And if you have a lot of prions in your test sample, you get very rapid increase in fluorescence. And this is a sum of 8 replicate reactions that we've set up in this well. When seeded with pretty concentrated, well, between 10 to the minus 3 and 10 to the minus 8th dilutions of brain tissue from an animal that has a prion disease called scraping. And it's only when you dilute that brain out, and we're only adding a couple microliters per each reaction here of these diluted brain homogeneous. It's only when you dilute it out to a billion-fold or so that some of the replicate reactions fail to become positive. So there, meanwhile, the samples seeded with a range of concentrations of normal brain do not give any reaction at all. So these assays are extremely sensitive, giving a billion or a trillion-fold amplification of these seeds here in the reaction. And with an immune capture step to concentrate these, we can even detect 10 to the 14th fold dilutions of human brain tissue from patients with variant CJD. That's the form of Creutzfeldt-Jakob disease that comes from exposures to BSE or mad cow disease. That can contain as little as 1 attogram, or 10 to the minus 18th gram of the abnormal PRP in that human brain specimen. So extraordinarily sensitive. We're down to almost the point of detecting a few particles, prion particles in this type of assay. It's almost like PCR, virtually like PCR in terms of sensitivity. They can also be quantitative in endpoint dilution modes or in other modes as well. We've designed them to be highly disease specific. And so far, they're faster, cheaper, and more practical than similarly sensitive tests that are out there. So we and a lot of other labs, in fact, I looked this up last night, it's now over 120 or so publications by various groups around the world who have adapted this, what we're calling RT-QuIC for real-time quaking-induced conversion test, to virtually all of the prion diseases that we know to exist in mammals. The human ones, both the spontaneous or apparently spontaneous sporadic Creutzfeldt-Jakob disease, the variant, as I mentioned, from BSE infections, various genetic prion diseases, sheep, scrapie, both classical and atypical forms, deer and elk chronic wasting disease, BSE in cattle, so forth and so on. And usually this developmental work is done with brain tissue, but that's not a very practical diagnostic specimen for sure. So also a lot of effort's gone into adapting these assays to more accessible diagnostic specimens such as those listed here. And what I'm going to focus on today are the applications to human prion disease diagnosis using cerebrospinal fluid, nasal brushings, or skin. And initially, the initial work on human diagnosis using cerebrospinal fluid for human diagnosis of human sporadic CJD using spinal fluid was performed by Ryu Atarashi after he went back to Japan along with his Japanese and Australian colleagues in this paper here. And since that time, we've collaborated with a UK group to perform similar large studies of the diagnostic utility of this sort of approach, and still other large independent studies have been published as well. And collectively, all of these large studies have indicated a diagnostic sensitivity of this approach between 77 and 89%. That is the percentage of sporadic CJD cases giving positive tests, and 99 to 100% diagnostic specificity. That is the percent of non-CJD cases giving negative tests. So this was really the state of the art. But we were, and really provided the first disease-specific diagnostic test that doesn't require either a brain biopsy or post-mortem neuropathological analysis. And as such has been implemented now by many major CJD diagnostic centers around the world. But we were still concerned about the fact that between 23 and 11% of the cases that were coming in the door were still being missed. I mean, this is, of course, bad for the patients and the doctors and families and so forth if they're not diagnosed properly. But of course, CJD is also transmissible, so the caregivers and clinical staff need to know. It's best if they know when they're dealing with a CJD patient to do all that they can to prevent any iatrogenic transmission of CJD to others in the clinical setting. So we set out to improve those conditions, prove on that sensitivity, and this was initially largely work of Christina Oru and Brad Groveman in the lab. And what we're seeing here then are traces, RT-quick traces as a function of incubation time here, averaging the traces from, in this case, 48 different sporadic CJD cases. They're cerebral spinal fluid samples, using the new conditions that these two came up with compared to the old conditions that were being used more or less in the previous studies that I just mentioned. And so you can see that we're getting, with these new conditions, so-called second-generation conditions, much faster and stronger responses. And we're also able to, in this initial study here, get positive responses above the threshold that we selected for 6 out of 48 CJD cases and no positives from 39 controls. And all of this has indicated that we're getting faster, stronger RT-quick responses that reduces the time required for running these assays from a matter of days down to hours. And if we combine these results here with a bunch of other samples that we have analyzed in my lab now and in our collaborators in Italy, in Gianluigi Zanuso's lab, in blinded testing, we're getting positive reactions from 152 out of 161 CJD cases, but not from 103 non-CJD controls. So this is giving an improved sensitivity up to 94% and still maintaining 100% diagnostic specificity versus non-CJD cases. So these new conditions are improving the performance and the accuracy and practicality of diagnostic testing for CJD. And we're not the only ones who can do this now, because my technician went to our national CJD pathology surveillance lab at Case Western, run at that time by Yishi Safar, and trained them how to do the assay. They then took off totally on their own and performed their own retrospective and prospective analyses of fairly large numbers. Again, this is a rare disease, so these are large numbers. Large numbers of CJD and non-CJD or non-prion controls, getting similar sensitivity and specificity to what we got in both of these studies. And so we were gratified by that. And then another large study done by Piero Parchi's lab in University of Bologna in Italy, another major diagnostic, prion disease diagnostic center in Italy, got very similar results as well. So we're not the only ones who can do this. It seems to be transferable, readily transferable technology. So so with the CSF, we're still not getting all the cases that come in the door. Another approach that we have since taken was the brainchild of an Italian neurologist, Gianluigi Zanuso. Brainchild, that's a bad choice of word. Who years ago published a New England Journal paper showing that CJD patients have abnormal PRP deposits lining the top of their nasal vault in what's called the olfactory mucosa or the olfactory neuroepithelium. And he asked me at a meeting, after hearing about our initial RT-quick reactions, if he devised a way to sort of collect a little bit of the mucosal layer from these CJD patients and sent them to us in Montana, would we test them in our RT-quick assay and see if we can pick up signs of prions in that sample. And these little brushings here collect mucus, but also some of the olfactory sensory neurons that give you your sense of smell up there at the top of the nasal vault. And these are normally sloughed off anyway. They have a half-life of 6 months or so. So this isn't a very big deal. And so he did that. He sent us fairly large numbers of brushings from CJD cases and controls. And this shows average tracings that we got from the nasal specimens from CJD patients versus CSF or cerebral spinal fluid samples from the same patients, in this case using the old rather than new conditions for CSF. But in any case, you can see these nasal specimens are giving much stronger and faster responses than the CSFs from the same patients. And then if you plot the THT, the maximum THT fluorescent signals from reactions run on the olfactory mucosa samples versus the CSF samples from CJD cases, you can see we're getting positive responses from more of them using the nasal specimens than the CSF specimens. So in this particular study of 43 total CJD cases and 43 non-CJD cases, we got greater than or equal to 97% sensitivity and still 100% diagnostic specificity, relative to, in this cohort anyway, a 79% sensitivity and 100% specificity using their CSF samples. So it looked like we were getting a better read on things with these nasal brushing samples, such that they seem to be capable of giving more definitive and, at least to some people anyway, a less invasive anti-mortem diagnostic test for CJD. And so based on those results, we went on and collaborated with Dr. Zanuso to look at what happens if you're able to use either CSF or nasal brushings, or if need be both, in diagnosing CJD cases. And this initial panel that we looked at involved 61 sporadic CJD cases and 50 non-prion cases. And overall then we were able to improve, using this strategy, the sensitivity to 100%. So we proposed an algorithm for diagnosing CJD in these patients. If you have a suspect CJD patient, probably the first thing that's going to happen, in most cases anyway, is that CSF will be collected, so you'll have that to test. Because CSF is collected from any rapidly progressing neurodegenerative disease case to rule out, say, an encephalitis or something that's actually treatable. If this RT quick assay is positive, then it's a probable CJD case and you're done. If it's negative, then you probably should collect, or you could collect, an olfactory mucosa sample and test that. If that's positive, it's probable CJD. And given these numbers here, if both of these samples are negative, you probably should reconsider the diagnosis, because it's probably something else. So as another backup, I mean, that's all fine and dandy, but in the US at least, it's hard to set up to collect those olfactory mucosa samples for certain FDA-related practical reasons. So it's always good to have still alternative diagnostic specimens that may be used under certain circumstances. And one of those that we've now identified is the skin of sporadic and variant CJD cases, which we've shown contain lots of this prion seeding activity that we measure in the RT quick assay, and also some infectivity that we can measure by bioassay in animals. So using skin specimens from these patients that have been collected either from the lower back, the top of the head, this is post-mortem of course, or behind the ear, we're getting strong signals from most all of those samples, but not from non-CJD controls shown here on the baseline. If you plot the maximum thioflavin T fluorescence from those various reactions, you can see we're getting, again, positive responses from most of the specimens. We don't always get a positive response from all locations where the skin was collected. But if you look at the overall score, putting together each of these different skin specimens, we've gotten positive responses from every single sporadic CJD cases, whereas not from control. So far, and these are small numbers, of course, in terms of diagnostic validation for sure, 100% sensitivity and 100% specificity if you're able to collect these 3 different specimens from each patient. So, and furthermore, as I mentioned at the outset, skin homogenates from 2 of the sporadic CJD patients were injected into mice that have been humanized to express the human prion protein gene and thus be susceptible to human CJD, and these transgenic lines, in fact, did then get infected and succumb to disease. So there's both infectious prions for sure and this prion seeding activity in these skin samples. So what are the implications of this? Well, we, of course, think it provides an alternative basis for diagnostic testing, at least post-mortem. You say, well, why would you bother with post-mortem? Well, it's a really important deal to get a clear diagnosis, even if it's only post-mortem for practical and surveillance reasons. And there's large parts of the world, my collaborators, Wen Quanzhou and Qinzhong Kong, have told me, and it's true in China and in India as well, where full-blown autopsies, especially of the brain, are not well accepted. And so it'd really be helpful to, even post-mortem, to take skin samples and be able to make a firm diagnosis. And we also think that it provides a basis for reports in now several papers that there seems to be some risk, epidemiological risk of getting CJD associated with having had prior surgeries involving skin, and in some cases only involving skin, as if there may be, through skin surgeries, a potential for contaminating surgical instruments and so forth that can then be transmitted. It's a low risk, and I don't mean to say the sky is falling. Nobody's saying it's a high risk, but if it's a risk at all, given how horrible CJD is for the people involved, it's worth considering further as something that might be real. So to summarize then, for these RT-QuIC assays for the prion disease diagnostics, we were able to get nearly 100% accurate ante-mortem diagnosis of sporadic CJD, at least when both CSF and, if need be, nasal brushings can be used. The skin, again, we've gotten positive reactions from all of the CJD and 2 variant CJD cases that we've looked at. We haven't published this yet, but in a collaboration with Christina Sigurdsson at UCSD and Michael Geschwind at UCSF, we've been analyzing all the different parts of the eye coming from sporadic CJD cases, and we're seeing all of them positive in our assays. So, and we've also, and I haven't told you anything about this, we have identified a universal recombinant PRP substrate that seems to be able to detect in one fell swoop every prion and every mammal that we know of in mammalian species, and that's coming from a little mammal called a bank vole. And we've learned that we can discriminate certain important strains, either in cattle or sheep and in humans, just by differential reactivity or behavior of these samples in permutations of RT-QuIC reactions. And importantly, in a practical sense, if you think of people setting up and running these reactions in a routine clinical diagnostic capacity, we find that the products of the reactions that we're amplifying in these plates, in fact, are not identical copies of the prions that are used to seed the reaction. They have all the seeding activity, but they're not faithful conformational templates. So if you inject those products of our RT-QuIC reactions, large amounts of it, into the brains of susceptible animals, they never get sick. So that's a practical benefit here. So the biohazard of running these assays appears to be low. You're not just cranking out massive amounts of infectivity every time you run a test. So let me switch. So that's the deal with the prion RT-QuIC. I now want to tell you about our efforts to apply this sort of technology to diagnosis of synucleinopathies, so-called because It's alpha-synuclein that causes the pathology in Parkinson's disease, dementia with Lewy bodies. And we've been working on this now for 3 or 4 years, and meanwhile, 2 other groups were working in the same direction to develop these sort of assays, Alison Green's lab in Edinburgh and Claudia Soto's lab in Texas. And basically what we all have been doing is to substitute monomeric recombinant PRP with monomeric recombinant alpha-synuclein, human alpha-synuclein. Same sort of scenario all along. And what the Alison Green's lab showed, published already before we got into press with our study, they got very good diagnostic sensitivity for both Parkinson's disease, taking their cerebral spinal fluid samples, sometimes pretty early in the course of disease, and also for Lewy body dementia, and 100% specificity with respect to non-synucleinopathy controls. Their assay time in this setup was about 5 days. Claudia Soto's lab got 88% sensitivity and a little bit different configuration of this sort of assay, really pretty high specificity. Their assay time as they published it was about 13 days. And so what we have to offer is a much faster assay that Brad Groveman in the lab developed. So whereas they're taking 5 to 13 days to do the experiment, ours is done in a day or 2. So that's a practical advantage, but I have to say our performance is pretty similar to that of Alison Green's test, in that we're picking up the vast majority, in excess of 90% of the Parkinson's disease cases and the Lewy body dementia cases, and not getting positive responses either from normal or otherwise non-synucleinopathy controls, including a bunch of Alzheimer's controls who have a different type of amyloid protein in their cerebral spinal fluid. Again, our sensitivity from this was 93%, and so far anyway, and we haven't done near as many samples as Alison Green and Soto have done, but 100% specificity. So it's all looking pretty good for all of the assays from all 3 of our groups. And we're all showing unprecedented diagnostic sensitivity and specificity for Parkinson's and Lewy body dementia. And I think I mentioned earlier in the talk that Parkinson's is misdiagnosed by GPs 50% of the time or so, until, almost till death. So this is really an important thing to do. Our conditions are dramatically reducing the assay time that's required. And this seeding activity seems to be a synucleinopathy-specific etiological biomarker. We have good reason to think we're measuring the disease-causing entity of these diseases. And this is really interesting, not just to diagnosticians, but to pharmaceutical companies as well, who are trying to develop treatments for these synucleinopathies. So they get more accurate, you can get more accurate and earlier diagnosis, so that you can improve your selection of your cohort of patients to put into a therapeutic trial. And they're also excited about the fact that you can do longitudinal measurements of this disease-causing biomarker in people who have either received or not received the treatment that you're trying to apply to these diseases. And the last part of the talk I want to devote to RT-QuIC tests for tauopathies, again, Alzheimer's disease, progressive supranuclear palsy, for instance, corticobasal degeneration, so forth. And this is largely, over the last 4 years or so, been very difficult but painstaking work, but now pretty successful, we think, by Ari Saijo, a postdoc in the lab. So just as background information about tauopathies, tau and tauopathies in humans. Humans normally make 6 different isoforms of tau due to differential splicing, and it ends up such that there are 3 isoforms with 4 of these microtubule binding repeats here, the 4R tau isoforms, and 3 with 3 repeats, the 3R isoforms. And different tauopathies, such as Alzheimer's, frontotemporal dementia, Parkinsonism, progressive supranuclear palsy, and so forth, can preferentially accumulate either 4 repeat tau isoforms, in this case 3 repeat tau isoforms, in this case in these distinct deposits that look like PIC bodies, they would call, and in Alzheimer's disease you get roughly equivalent accumulations of 3 repeat and 4 repeat isoforms. And so what Eri Saijo did initially, because it turned out to be what worked first, was to develop an assay that was really optimized for a 3 repeat tauopathy called PIC disease. And this is showing RT quick responses. Each panel are 4 replicate reactions seeded with serial 10-fold dilutions of PIC disease brain, all the way out to 10 to the minus 9th, 10 to the minus 10th. And you can see that all the way out to 10 to the minus 7th, you're getting, I think that's all 4 replicate, yeah, it is, all 4 out of 4 replicate reactions giving positive responses. It's only when you're way out to 10 to the minus 8th, 10 to the minus 9th, or billion-fold dilution of this PIC disease brain that you're starting to lose positivity in the replicate reactions. And then negative reactions from non-tauopathy controls with much higher concentrations of brain. So these conditions she established detect 10 to the 3rd to 10 to the 9th fold dilutions of human PIC disease brain homogenates using only 2 microliters of them. So again, extraordinarily sensitive detection of the misfolded tau particles in there with seeding activity. And so this represents sort of an endpoint dilution, and you can do a Spearman Carrboro analysis that allows you to calculate for a bunch of different specimens basically the concentrations of seeding activity that are relative concentrations of seeding activity that are in different types of brain. And these are plots then of on a logarithmic scale of the log of these seeding units per milligram of brain tissue. For PIC, a whole bunch of 8 PIC disease cases shown here versus brain samples from Alzheimer's, another tauopathy, and other tauopathies here as well, PSP, CBD, AGD, so forth and so on, and then a whole bunch of disease cases that have no at least immunohistologically detectable tau deposits at all. It doesn't mean they don't have any, it just means it's not detectable by immunohistological examination. But in any case, you can see extraordinarily, many logs, let's say 4 to 5 logs, higher seeding activity for PIC disease with this assay than even the other tauopathies, despite the fact that these other tauopathies, if you look biochemically, have pretty similar levels of abnormal detergent-insoluble tau aggregates in those brains. So there's extraordinary selectivity to the seeding here for this particular type of tauopathy called PIC disease. And she and Alison Krauss and Mike Metrick have gone on recently in the lab, this is data that I just showed you, to develop other types of tau-RT quick assays that are optimized for other types of tauopathies. For instance, out over here on the right, they've got one that's optimized for 4R tau, for certain 4R tauopathy called progressive supranuclear palsy. Here you can see you get some of these, one of these cases gives you, can be diluted all the way out to 10 to the minus 9th, 10 to the minus 10th, and still give positive reactions here, and others still give strong responses that in all cases are logs higher than the responses you get even from other 4R tauopathies. So it's extraordinary selectivity and specificity to the seeding activity that these different types of tauopathies have. They've developed one, they've optimized an assay for sporadic Alzheimer's disease shown here, and in this case, under these conditions, there's more promiscuous detection of seeding activity and a whole raft of other types of brain diseases. Relative to our absolute rock bottom negative control is shown here in these little boxes at the bottom in this line here, which is actually what you get from a totally tau-free, tau knockout mouse brain. So we're trying to figure out what to make of these levels of tau seeding activity. It's readily possible that in all of these different diseases here, the neurodegenerative diseases here, that you get some accumulation, albeit at much lower levels than we were picking up for the disease for which we've optimized the assay in those brains, maybe as a comorbidity or something. So we have profound selectivity for each of these different types of tauopathies over others. So to conclude then, for the tau-RT-quick assays, we've got several assays, in fact 2 different ones for the 4 repeat tauopathies with different selectivities that can detect down to billion-fold dilutions in some cases of diseased brain. And each assay can have orders of magnitude selectivity for the type of tauopathy for which it was optimized, which may in fact explain the replication of multiple distinct tauopathy strains. So Mark Diamond's lab, for instance, at UT Southwestern has isolated 18 different types of tau aggregates in cell culture and then in transmissions to transgenic mouse models that faithfully propagate in cell culture, giving tau aggregates with reproducibly or consistently different biochemical characteristics, and then when inoculated into animals, give them different pathologies, different distributions of the misfolded tau, so forth and so on, that are behaving a lot like prion strains behave, real PRP-based prion strains. So this profound selectivity may explain how that's possible in vivo. And we have been able so far to pick up, this isn't too exciting yet, we've been able to pick up seeding activity in post-mortem CSF samples collected from Alzheimer's and PICS cases, but post-mortem collection of CSF is not like ante-mortem. We think there's some brain contamination that probably happens when you do it post-mortem. But in any case, we can pick up seeding activity in the matrix, cerebral spinal fluid at least. And all of this provides a potential basis for more accurate tauopathy diagnosis. And again, as I said, for the synucleinopathies, better cohort selection and biomarker measurement in therapeutic trials. So over the long haul, our fantasy, and hopefully it's more reasonable than a fantasy, is that we and others can develop a large panel of different RT-quick-like tests, seed amplification tests, that can detect and discriminate these different types of protein misfolding diseases, so that once the patient first shows up in a clinic with the earliest of signs, and you have a reason to suspect it might be one of these degenerative protein misfolding diseases, you could apply this large panel of seed amplification tests to them and tell them what the molecular pathology or pathologies is. Sometimes, as I mentioned, it's a combination of misfolded proteins, like in Alzheimer's, amyloid beta and tau at the same time, and this happens quite a bit. You can tell them which molecular pathologies they have, so that you can get started with treatments as early as possible that actually have a chance of working as they come available. And so, yeah, again, the earlier you can start, of course, the better the prospects for treatment, once treatments are available, but also just in the development of treatments as well. So with that, I just want to make sure everyone understands that there's just a massive number of people who have been involved in doing the work. Up here in my lab, bolded here, I've mentioned these along the way for the most part, but also a whole slew of clinical colleagues like Gianluigi Zanussi at the University of Verona, Maurizio Poccari at the ISS in Rome, Bernardino Getty at Indiana University, Doug Galasco, Christina Sigurdsson at UCSD, Michael Geschwind at UCSF, and a bunch of people at Case Western who have been extraordinarily helpful in getting us the diagnostic specimens that we need to do this work, which I think is probably the hardest part. So with that, I will stop and take any questions that you have.
wondering how deep into the skin we need to go to get a sample, and what the specific infectivity is in skin, and is there a possibility of shedding through sloughing off of dead skin cells?
Right, those are really good, important questions. So far, we've been taking punches a day. This is actually, and I failed to emphasize this enough, is the skin work project was spearheaded by Wen-Quan Zhu at Case Western, and he's the one who has been sending us all these samples or extracts of skin samples. But they have been taking full thickness punches and sending them to us. And so we really don't know at this point what part of the skin it comes from. We assume, based on what's been seen in hamsters anyway, and I think sheep, but mostly hamsters, is that may be in the nerve endings and things that are there, but maybe not exclusively. We really want to know, of course, whether this material can move to the outside of the skin and slough off in any capacity. I mean, at this point we have and you asked about the relative, the specific infectivity in the skin. It's 1,000 to 10,000 fold lower or so than what we see in brain. So it's much lower. And so what might be on the outside of the skin could be quite a lot lower still, and may simply not ever be enough to represent a biohazard or any risk of transmission by casual contact. I mean, we want to look into it. Well, Brian Appleby is collecting, at Case is collecting, setting up to collect anyways, some scrapings from the surface of the skin so we can address these questions. But they are really important. Did I answer all your?
Yeah.
Okay. Yeah.
So during your talk, I was thinking about how conserved are prion proteins between species, because since there's no transmissibility, but I think your RT-Quick kind of explains that even though they're very conserved, the specificity of this amplification is incredible. But I'm puzzled because I think at least there was one report where they showed that alpha-synuclein fibrils could promote beta amyloid aggregation. So in that case, it's a case where there's not that much specificity, and how can you explain that?
Well, I mean, it's a very important issue what you're bringing up, is this possibility of heterologous seeding between different types of amyloids. Now, we've explicitly tried to derive conditions that do not allow that or promote that. But that isn't to say there aren't conditions, even in the body, that allow heterologous seeding to happen. We just are not going that way. Well, it depends. With our bankful assay, we were going for, very universal and non-selective seeding for different prion ones. So you can just set it up with your conditions and your substrates and cofactors and all that sort of stuff to either focus on selectivity, focus on sensitivity, or not. And so one possible way of thinking about it is not if this is a fibril and you've got the seeding surface on the end. There's a couple things that can happen. I mean, most of the time we're thinking, as I've drawn in these cartoons, seeded polymerization, templated polymerization off the ends. So this structure is imposing its conformation on each monomer that joins. But there's another kind of nucleation that happens that almost has to happen to explain the kinetics of what we're getting, and that's secondary nucleation, which we think involves sort of the lateral scaffolding of at least in the case of PRP molecules along here that then make their own amyloid. And we don't even think it's necessarily the same template as this amyloid, but it's happening because of alignment along the sides of the fibrils. So it's even possible that if you have, say, an alpha-synuclein amyloid like this, that it may somehow, because it's such a nice repeating surface of amyloid, might promote the assembly of amyloid beta peptides along there, or some other, or prion protein, or some other amyloidogenic protein along there in a secondary nucleation way that could be problematic in our assays, or could be an issue in vivo. And in fact, Claudio Soto has published recent papers which suggest heterologous seeding in vivo between the amyloid of type 2 diabetes, that is amylin amyloid, IAPP amyloid on one hand, and Alzheimer's beta amyloid on the other. In other words, if you inject pancreatic extracts from diabetics into their transgenic mouse model of Alzheimer's disease, you can exacerbate Alzheimer's pathology, and I think vice versa. Don't have all the details in my head, but this is a relevant issue you're bringing up, whether it's due necessarily to direct interactions between the two types of amyloids, or whether it's the fact that one type of amyloid can overload the protein quality control mechanisms in the body to allow the spontaneous accumulation of the other type of amyloid is a whole nother issue. But I mean, there's some evidence there's direct interactions. But all of those issues are important, not just for practical point of view, but from physiological point of view of understanding these pathologies and how they may work with each other, especially as we age and our protein quality control mechanisms start to break down. Yeah.
Could you comment, you had mentioned briefly, very nice talk by the way.
Thank you.
Briefly on the longitudinal sampling, could you give a feel for when misfolded proteins might first appear in the CSF to be available for diagnosis? We know these diseases have a long asymptomatic period, so when do they start showing up, and then is there an increase during the clinical course from, say, early onset to late onset of disease?
Well, to answer your last question first, we're not seeing a sign of that. And we don't know the answer to your question when it comes to humans. But people are starting to address that. Some of these clinical centers who have access, especially to familial prion disease cases, you're not going to get preclinical specimens from people who are not familial prion disease candidates, right? Because they're not going to show up in the clinic and want their spinal fluid taken without a reason. But if you have a familial, if you're from a family, or you've identified families that have a familial prion disease mutation, then you can set out to test exactly what you're asking about in humans, but we don't have the information on that yet. We do know that we've done in my lab longitudinal studies of CSF in scrapie-infected hamsters, and it shows up a little bit before clinical disease, but not much, almost at the same time. Whereas in skin, it's showing up way before. It is going to depend, of course, on how you inoculate. The route of inoculation is scrapie into the animals, but in the skin, we're seeing it many weeks before they actually get sick. So what the story is in humans in that scenario, we don't know, but it's really important to figure out. Any other questions? Yeah.
For your false negative samples, do you know what it was about those samples that made your test not detect the protein?
Are you talking about prion disease or Parkinson's, say, or?
Any and all of them.
Okay. In the Parkinson's case with the alpha-syn RT-QuIC, I'm not sure of all the reasons why, but the clinicians that I've talked to, and Doug Galasco, the clinician at UCSD who sent us this blinded panel of samples, some of which collected, or many of which were collected early in the course of Parkinson's disease, he was actually happy to see that we only picked up 93%. It's something that clinicians are expecting, and in part due to the fact that at least that percentage of the time the Parkinson's disease is misdiagnosed, so some of these patients are still alive. Or it may just be that 7% of Parkinson's patients just don't end up with any alpha-synuclein that's measurable in their CSF. We don't know that, which it is. And I guess I'd have to give the same answer for, well, in the case of the prion disease thing, we know it's not a misdiagnosis because they've all been neuropathologically confirmed one way or another post-mortem. But in the Parkinson's cases, we don't always have that information. So either we're not sensitive enough or it's not there. You know, we've got to figure that out with much larger numbers of samples as time goes on. Anything else? Okay, if not, thanks.